maybe scroll up here. Okay. So let's talk a little bit about what are the actual products from the light reactions. And then today we're going to start on how they're actually used. So we have this fairly complex system, and it's usually discussed of as having these four major components. And you'll see similarly arranged components in the mitochondrial membrane. So we have two locations of where light energy actually enters in the system. PS2 and PS1. And that these things are arranged in the membrane in a very particular vector way, so that the electrons, when they're moving through here, when they're moving through these oxidation and reduction reactions, they are putting protons into the lumen, inside the lumen, and then these protons are going to move back through the ATPase, and roughly what we think now, and remember this thing actually spins around, is about every time we have 3 protons that move this, then we get an ATP produced. So the ATP is produced, and then the electrons then actually are used to reduce NADP to NADPH. So now we have some stored energy here in the forms of this reduced NADPH. Okay? And what we're going to start talking about today is how these are used then to fix carbon. And then we have ATP, and ATP then is used to make a lot of reactions happen. And in a lot of instances, the phosphate from the triphosphate, the phosphate will actually be added to the product. So you have ATP plus product plus substrate, ADP plus substrate with a P attached. Sometimes with a P attached, sometimes not. And so we're going to see this energy is moving around. Now, back to the home page for the class. Down there's a review of chemistry. So if you've not looked at those reviews, there's a review on oxidation reduction. There's a review on basic biochemistry. If you click on that, it takes you to, if you're familiar with the Khan Academy, where you can get a good review of the biological nature of these reactions. Okay, it's a big difference. We're not just doing something in a mixture. This has cell membranes around it, and it has a whole series of reactions, and each reaction requires the presence of an enzyme. So these things get very complex quickly. Okay? Now, we finished up with talking about the herbicides, and we finished up with the fact that both the chloroplasts and the mitochondria have this same way of producing ATP. And the cycle here is, of course, the protons moving through here, and then the protons through the series of these very complex reactions of, here we see the protons moving in, okay? So we're pretty sure that these things came from the same place, obviously, over time. Okay, questions about that? Okay. So now we are going to talk about the dark reactions. Okay, so this is what we're going to be doing today and then Wednesday. Friday there will be no class since you'll be setting the exam that day. And we're going to talk now about what's called the dark reactions because these do not directly require light. The only thing light is required for is what? What are the products of light reactions? I just went over them a minute ago. NADPH and ATP. So that's the light reactions. Now, of course, these things are coupled together, obviously, because we're moving electrons around now. We're moving compounds around. We're oxidizing and reducing compounds. But the light reactions produce two things, NADPH and ATP. Oxygen is just a useless byproduct in terms of energy, okay? Now, obviously, when I say that, over time, plants and then animals evolved to use oxygen, and you're going to see where oxygen is actually used. But initially, oxygen, when all this stuff got started, there probably wasn't a lot of oxygen in the atmosphere. So oxygen over time then, first whatever these pre-plants were, and then animals evolved mitochondria to be able to use the oxygen. Now, we're going to talk about what everybody knows about the Calvin cycle. It's also called the C3 cycle. There's a C2 cycle, there's a C3 cycle, and there's a C4 cycle, and they're called different things. And I'll try to stick to one terminology. And we're going to start off with what most plants have, which is the C3 cycle. We're going to spend a little bit talking about how the CO2 gets in, which is, of course, integrally related to water potential and the stomas opening and closing. And then we're going to talk about something called photorespiration and how plants can get around that. Now, we'll then talk about the modifications that plants have evolved to survive under different kinds of light and different amounts of water. Okay? Oops. Now, this is where the stuff starts. This is the stomate. And this is where water is coming out, because that's the only way water moves up plants, is that, where's the lowest water potential? Between the leaf, stem, and the air, where's the lowest water potential? The air, okay? So that's driving the movement of water out of the stomate. Okay, so we got a plant, it's got plenty of water, stomates are open, it's losing water, and as it's losing water, CO2 is going in. And there's all sorts of different kinds of stomates. The stomate basically is composed of these things called guard cells. The opening is actually called the stoma, the hole. And then what's really important, and you've probably heard of guard cells, but you may have not heard about subsidiary cells. And these are the cells that are next to the guard cells, and this is where all the biochemistry goes on that helps the guard cells open and close. And let me tell you now, we still fully don't understand what causes these cells to open and close. These kidney-shaped cells open and close because water is going to flow in or out. Okay, and we're going to talk a little bit about the water potential of guard cells. When will these guard cells be closed? Under what conditions?
Nighttime. Nighttime, but
under night they can close some. What other conditions will cause the stomates to close? What other?
drought.
Low water? So low water means you want to close these things down so you don't lose water. So what does that do to photosynthesis? fixation goes down. So this is something that we're always fighting with in terms of making plants grow better. There's this grand challenge that the administration has come up with, or the dean, not the administration, one of the deans. Water use efficiency, making plants more water efficient, and the thing to do is to use these molecular markers, and he doesn't have a clue what he's talking about. It's a very complex thing. You just can't jump into the end. You've got to create population after population of plants that have different levels of whatever water use efficiency is, drought tolerance, and then you just can't jump in at the end and use all these multiple markers to select for the plants. It's a very complicated thing. And the notion that a plant can become more efficient in using water is very, very, very hard to do. A plant has the same productivity. The plant's going to need as much water as it needs. Okay, so we don't know any magic genes yet. The hardest, cloning or making a plant, putting DNA into a plant is easy. Figuring out what DNA to put into a plant is hard. So you have a population of plants. You plant a population, and you start making selections. Maybe you're going to be in South Texas where the salt levels are higher. So you're selecting for plants that grow better with higher salts. That doesn't mean they're drought tolerant. That means they may have some way of putting the salts into the vacuole, which will lower their water potential and allow the salty water to come in. So there's a whole lot of things. But we don't know. We don't know what makes a plant drought tolerant. But right now, what we're talking about is increasing efficiency is by decreasing the amount of water we waste, right? What else can you do? We don't know of a gene or a series of genes that we can transform into a plant to make it magically use less water. And probably we'll never be able to if the plant is going to have the same growth and yield. When I say never, I mean not in the next few years anyway. So we have all these controls that we have. Most of the stomates are on the lower epidermis. Of course, some species they're not. And we've talked about the guard cells, and there's this stomatal complex, which is the whole series of cells and cell types that are needed to make them work. Now, this is a nice diagram and a picture of a stomate. And one thing you'll see that's in the guard cells are two things. First, you'll see chloroplast, and then you see these little tiny things here are actually probably part of a vacuole. And then we have the cellulose microfibrils that are attached in this fashion here. And this is what helps the guard cells to open. Let me say, when they close, they're going to be flat here in the center. And so what happens? So what makes these things open and close? Well, guess what? It's water potential. It's water movement. And this is from your book. And because of the way the microfibrils are arranged, when they do take in some water, then they're going to swell up. And because of the arrangement, and you can do this, there's a really neat, someone sent me a really neat YouTube video too about a rap about water potential, but there's another one I'll try to find where you can take balloons, just take normal balloons and wrap them, put tape around them, not, just strips of tape around them, and you start blowing up the balloon, then the balloon will deform one way or another. So that movement, this radial movement, causes this opening in the center. Does that make sense? Okay? And then when the water then moves out the other way, these things close. So the last point of moving the guards, or opening and closing the guard cells, is the movement of water. It's water potential. The things that control the water potential are not completely, totally known. Questions? What moves around? Well, we know a little bit about what moves around. We can have chlorine move in, potassium move in, and as these move in, probably with some transport, so a proton moves out, then that's going to increase the solute potential. It's going to make the solute potential more negative, okay, more negative, and then that's going to cause water to move in, right? And then the thing starts warping, and you have it opening. Okay? Let me go in here. The way these things are regulated, the absolute way these things are regulated, is still not quite completely understood. So we know more about how the salts move in than we know about why the salts move out to close it. And we can get these things to change incredibly quickly. So there are some experiments that if you take a plant and you very quickly stress it, then the stomates will actually close before the water has actually moved, before whatever would be in the water, a signal in the water, has moved from the roots to the leaf. So in other words, it's happening faster than whatever could be in the water that's moving up. So there might be some other kind of signal, hormone signals. Hormone signals can move faster. So in other words, you stress a root, the water in the root, then let's say add some hormone to it, and that hormone moves up as the water moves through the plant. Okay? And there's a time for that. But what has been reported is the stomates close before that whatever was in the water can move up. So we're probably having something transporting, active transport, you know. You've probably heard of ABA. ABA will cause the stomates to close. You can spray ABA and you'll cause the stomates to close. But we don't know as much about how they could, and controlling the stomates closing is just not a stopping of the pumping of the ions into the vacuole. It's that they're also going away. Okay, so we know less about that. Questions? Okay, now we talked about stomates and how important those holes are in order of getting CO2 in. So we're still talking about, to a degree, boundary layer. Remember the boundary layer, which is that little, just a small space right above the stomate on the leaf. And if there's a lot of wind, then the water's going to move out faster. Okay, and that may actually even decrease a bit how fast CO2 can move in. And we're going to talk about now how these things, how plants take CO2 and reduce it finally to carbohydrate. It is the reduction cycle, PCR cycle. It is the Calvin cycle. And what is amazing is all of these reactions occur, and you're going to see how complex the PCR cycle is, one CO2 at a time. So this entire cycle occurs for every single CO2 that's added. Okay? Another new term that we will start talking about is, oh goodness, here we go again, is triose phosphate. And triose phosphate is a generic term that we use to talk about 3 carbon compounds that have a phosphate attached to them. And you'll see Wednesday and next week that triose are sort of the center part of metabolism. Now, if we look at this from the standpoint of what's going into each phase of the cycle, we have carboxylation. Okay, carboxylation is the reaction that adds the CO2 to a carbohydrate. That's the actual CO2 fixation reaction. Then the next phase we have is a reduction phase. It's reduced. So this is where most of the ATP and NADPH is used. So where do the ATP and NADPH come from? The light reactions. Then most of the reactions are actually regenerating the starting product, which is ribulose bisphosphate, RUBP. And then there's some ATP used in this part of the cycle. And then the RUBP and the CO2 then starts the cycle again to produce 3-PGA, PGA, phosphoglyceric acid, which you just need to remember is triose phosphate. Okay? So let's look at these reactions as they occur. The first phase here is one reaction. RUBP bisphosphate plus CO2 is converted into two 3-carbon compounds. Okay? This was the mystery reaction. And this is why it's called the Calvin cycle, because the scientist named Melvin Calvin was the person that figured out how this reaction occurs. And he was in California, and this all occurred right after the end of World War II. He was in Berkeley, and what did Berkeley have that you couldn't get anywhere else? Anybody know? Radioactive compounds. Berkeley was involved in developing the bomb. So there were a lot of people that wanted to do this experiment, but Calvin just happened to be at the place where he could get radioactive CO2 or make radioactive CO2. In 1946-47, you couldn't order it. You had to make it. And there was very few places. Berkeley and Oak Ridge was the only two places that you could get radioactive compounds to research. And so he was able to follow, using radioactivity, he was able to follow what compounds were produced. So this first reaction, the carboxylation reaction, is just a CO2 being added. Okay? Then the second part, which is in this red box, is where most of the light products, the products from the light reaction, are used. Then the remainder of this is used to regenerate the starting material. So all these reactions are used to regenerate the starting material. And also, we'll talk more about this a little bit later, but every time this cycle moves, every turn of the cycle, we're adding one CO2. So after 3 turns of the cycle, we have 3 new CO2s. We have one 3-carbon compound. And as it turns more and more, we start building up more and more carbon. And then that extra carbon, so first that's going to happen is this is going to be just completely, the cycle is going to be regenerated. That extra carbon then flows through a tremendous number of reactions to end up producing starch or glucose. So we're getting fructose, then glucose 1-phosphate, and eventually starch. Okay? Starch is produced when there's a lot of light, and this thing is going like crazy, and it's producing a lot of, it's adding a lot of CO2 to the cycle. The reaction here, let me go back, sorry. The reaction here, the carboxylation, Rubisco, RUBP, bisphosphate, this protein, the protein that catalyzes this reaction is the largest amount of protein in the world by weight. So think about this. A tree in the leaf is fixing a lot of carbon, but it's fixing one carbon at a time, period. Every time that cycle turns, every time these reactions occur, you get one extra carbon fixed. That's a lot of light reactions that are needed for the dark reactions to occur, okay? That's pretty amazing. You guys know what C4s are. We're going to talk about them in a bit. But these plants, C3 plants, don't grow as fast as C4 plants. So we have plants that we're going to talk about that are adapted to a lot more light. But this is pretty amazing. Every time this occurs, one CO2 is added. Okay? Questions? Okay. Now, let's go through each phase, or the main phases. The enzyme, the short-term name, which everybody calls it, is Rubisco. And what happens is, and this is what was so hard to find, how do you go from this to a 3-carbon compound? Well, that's because when CO2 and RUBP is added, it produces, for just a length of time at all, a very unstable 6-carbon compound. And then as soon as this 6-carbon compound is produced, it's so unstable it breaks down to 2 3-carbons. And that was the big question. Okay, I've got CO2, and it looks like this 5-carbon compound is involved, but the product is a 3-carbon compound. Doesn't make any sense. Well, yeah, 5 and 1 is 6, divided by 2 equals 3. And you're going to see that in a lot of reactions where 6-carbon compounds like glucose are immediately broken down to 3-carbon compounds. Then the first stable product of these reactions is 3-PGA, or triose phosphate, is fine. Here's the actual reaction that occurs. If we add radioactive CO2, so this little asterisk is supposed to be that it's radioactive, then when this reaction first occurs, we get this very unstable intermediate, which is then very quickly converted into these 2 3-carbon compounds, with one of them now being labeled, okay? Then in the next phase, we have, and you don't need to worry about these intermediates other than that they are used then to reduce these compounds, okay? And if you're familiar with dihydroxyacetone phosphate, we're going to be talking about that a little bit later, and glyceraldehyde 3-phosphate. And then in the regeneration phase, 5/6 of the triose phosphate is used to regenerate this pathway. So what does that mean? 3 turns for 1 triose phosphate. We have 6 turns to produce 1 hexose, one glucose, and then 12 turns to produce sucrose. 3 turns of the cycle, we need 6 NADPHs and 9 ATPs. So we use a lot of that light energy to reduce these compounds. Again, it's just a series of oxidation reduction reactions. Okay. some things about light and dark reactions. In the dark, leaves are taking up oxygen and giving off CO2 through respiration. In the light, it becomes much more complex. We have O2 being taken up and CO2 being given off. We have CO2 going in to the cycle. We have O2 coming out, and then we have the reduction cycle. So we have, in the light, we have CO2 still going in for fixation, but CO2 is being given off through respiration. So mitochondria are producing CO2 and using O2, and we'll talk about that when we talk about respiration. Chloroplasts are giving off O2, taking up CO2, and we have all these competing reactions, okay? At night, it's pretty easy. We only have O2 going in and CO2 coming off. So a lot of times people will measure that respiration rate at night and then come back and try to measure it in the day, and it's much more complex. Now, you've seen this several times, and I told you you'd see it again, and this is the again. We have a major problem with carboxylase, Rubisco. In the real world, outside, now, oxygen limits photosynthesis. Oxygen limits CO2 fixation. In the real world, this isn't something we do in the lab. This is what's going on out here. And here's the curve you've seen, I think, 2 previous times. And what we find is that if we increase the oxygen level, we decrease the efficiency of photosynthesis. Okay? That's because this enzyme, let me go ahead and start here, is that the enzyme Rubisco also can bind oxygen. Rubisco, which is the largest protein by weight, and it's what grabs CO2 and turns it into the stable triose phosphates, every once in a while can grab an oxygen. And if you grab an oxygen, there is no net increase in carbon, right? So this is going on out there now. Okay? It's real. It affects the yield of C3 plants. We haven't talked about C4s. But what happens is 2 things. Well, it's one thing, but it's 2 results. If we add oxygen to the system, we end up with 5 carbons. Okay? If we add CO2, we end up with 6 carbons. So we end up with a net increase in carbon. With O2, we end up with only 5 carbons. We start with 5, you add an oxygen, you're going to end up with 5. Okay? And this is called several different things. This is called the C2 pathway. It is also called photorespiration. Have you heard of photorespiration? That's the term that's used to describe this thing that happens maybe a quarter of a time, or less, maybe a little less than a quarter of a time. So the yield out in the real world is limited by this. It's real. And you know about C4s, we're going to talk about C4s probably next time, but if I go out in the summer in front of the building and where there's not any irrigation, and it's not rained in a while, and there'll be a lot of plants that are dicots that are wilted, and there'll be other dicots that are perfectly fine. I know that plant that's not wilted is a C4, and the plant that's wilted is a C3. And we'll talk more about that when we talk about C3s and C4s. But this is a real problem. It's not a problem, it exists. And it involves 3 different organelles, the chloroplast, the peroxisome, which I don't think you've heard about in the mitochondria. It's called the PCO cycle, the C2 cycle. I just prefer to call it the C2 cycle. And what happens, and you just need to worry about the fact that it occurs in these three organelles. And basically what it does is it fixes oxygen, okay? And produces all these intermediates and can even at some point produce CO2, okay? So you can even lose CO2. So you have a carbon that's already fixed, and now you're going to peel one off and turn it off to the atmosphere. Okay? People have tried. There has been a tremendous amount of work where we take Rubisco and we take that active site and we see if we can modify the active site so that we'll only see CO2 and not see oxygen. Because if we could do that, then plants will become much more efficient. And this is, it's not a real world problem, it's a real world thing. This limits crop productivity. We're going to talk in a bit about what does the increase in CO2 in the atmosphere do. There's been, well, if we increase the CO2 concentration, it's a competitive inhibitor, right? So as the concentration of CO2 goes up in the atmosphere, photorespiration should go down. And they're still trying to figure that out. And we're going to talk about some of these experiments they're doing out in fields where they're regulating everything. Okay, questions about this? The ratio is about 3-to-1. And another thing that photorespiration results in is the loss of CO2. So not only do you not fix the CO2, you lose the CO2. Okay? Why then has this pathway been maintained? Okay? The answer is nobody really knows. There's a lot of ideas. Here's some of them. You get some amino acids produced. There is the notion that this may occur when plants have a lot of sunlight hitting them, and they can peel off some of the ATPs and some of the NADPHs by going through these reactions. There's also an idea that photorespiration may keep the level of oxygen around what it is in the atmosphere. Have you heard that one? And because as the oxygen levels go up a little bit, or a little bit more, then actual oxidation becomes a problem, like in rust, this kind of stuff. Actually, if you get above the level of oxygen in the atmosphere, then we get into where systems there's too much oxygen. There could be too much oxygen. So there may be over time this has been maintained sort of to keep the oxygen levels where they are. I don't know if any of those, that's my favorite one. I like that. It's sort of bizarre, but that makes sense, I guess. But we don't really know. You will read why it's there, but we really don't know. There's only been one person that was able to separate the oxygenase from the carboxylase. The guy's name was Mark Spector. He did it for his master's degree, I think at Ohio State. He then went on for his PhD to cure cancer. And then it found out all his work was, of course, faked. He went to Cornell for his PhD, and his work was so outstanding that the chair of his committee, who was a distinguished professor, retired so that Spector could take his chair. So if you're going to cheat, don't separate the oxygenates. Don't do something that's so earth-shattering as, to come up with a Rubisco that doesn't see oxygen or cure cancer. Somebody's probably going to check up on those things. And with that, I'll stop and see you on Wednesday.